Hello there. We're going to start now that we know what we're talking about. After the sugar notations, we're going to see how we actually metabolize some of the sugars that we find in our food. Uh, and we're talking about here other than glucose. So we know what happens to glucose. So the point is to feed them at various points in glycolysis. So galactose joins way up. Galactose is the first one we're going to talk about. We have a few more. And it joins way up at the glucose 6-phosphate level, and it's actually by far the most complex. The rest are fairly straightforward to utilize. So how does galactose look like compared to glucose? Again, they only differ in one stereocenter right here. Okay, this one and this one. This carbon, so 1, 2, 3, 4 carbon 4. Very similar. Okay, where do we find it? Well, it's in the milk. It's also called milk sugar, and we get it in every dairy product. It's a so we don't get free galactose. We get lactose, which is a disaccharide. So it's got two sugars, glucose and galactose, and then it has to be broken down, and then we can use, of course, the glucose. So lactose is broken down, and then we get each monosaccharide separately. We get glucose and galactose. We know what happens to glucose. Let's see what happens to galactose. It's obviously a great source of energy. It's carbohydrate rich. Um, but let's look at lactose first. So this is how it looks like. It looks like this part is galactose, and this part to the right is glucose. So they are joined by a 1,4 glycosidic bond. Um, the hydroxyl group at carbon 1 of galactose is in the beta configuration. It's a beta anomer, and we're going to describe anomers in a little more detail in a minute. So it's this bond. The point is, it's this bond that has to be broken before anything can happen, right? And if this bond is broken enzymatically, then we're going to get galactose and glucose. And the enzyme, of course, that does that is lactase, which you can buy at the grocery store. And it's called lactate and the generic versions of it. All right. And why do you take it? Because if you don't make that enzyme anymore, so it happened to me a few years ago. So I reached that age where I lost my lactase, I guess, my general one. So it cannot be broken down, and then it gets metabolized by the bacteria in the gut, and you've got all kinds of enteric problems. Those of you who are lactose intolerant, and I'm sure some of you that are watching this video are almost certainly lactose intolerant, you know exactly what I mean. So, all right, so it's broken down. I mean, all mammals, when I mean, that's a definitive feature of mammals is that they lactate, they make milk, and babies that lactate. I mean, they of all kinds of mammals, they do have lactase, and they can break it down. So the point that we're going to discuss here is, oh, by the way, so just before we go on how galactose is metabolized, so as I said, mammals that make it like cows, you know, now we can get the lactose-free milk. There is no mutation there, because I get this question a lot, actually. There is absolutely no mutation in the cow. There is no transgenic cow. It's the same cow that makes the same milk. But when the milk is taken from the animal, they just add lactase in it. So it already comes with the enzyme added. So it's already broken down. All right, so let's see what happens to galactose now. All right, so we have a beautiful pathway. It's a very exotic, very unusual. You're never going to see anything like it, actually. You're going to see parts and pieces of it. And to be honest with you, as we'll see, there is no obvious reason why the pathway is the way it is. It's a little more. It seems a little more complex than it should be. But it was figured out by Leloir, who got the Nobel Prize in chemistry. And if I'm correct, he was the first chemist Nobel Prize from Latin America. He was from Argentina. So this is the overview of the pathway, and we're going to discuss its reaction in detail. So we get galactose, it gets phosphorylated at position 1, okay? And so we consume an ATP there, just like we do for glucose, let's say, when we bring it in. But that's then we have a lot more things to deal with. Um, in an interesting reaction that we're going to discuss, um, well, by the way, galactose, what is not shown here, some of the conformations change. Okay, so we have some extra enzymes to make sure that you go from the beta to the alpha anomer. So it can be worked on by subsequent enzymes, the galactose that is. Then furthermore, you have a reaction that we have not seen before, but we're going to see a lot more from now on, which is it reacts with activated glucose, and we'll see how glucose gets activated in this way, in an interesting way. It gets utilizated. We're going to explain what that is. And then you end up you swap the two, so you end up having, so when you start with activated glucose in the form of UDP glucose and galactose 1-phosphate, what you end up with is glucose 1-phosphate and UDP galactose. And that can be recirculated back into UDP glucose fairly easily. So that thing keeps going. Once you activate once glucose, then it can be recycled every time you use one galactose. So there is some energy input here, but it's only once. You can keep doing it again and again. Then once you get glucose 1-phosphate, what you have to do is to convert to glucose 6-phosphate. I mean, glucose 1-phosphate is not a glycolytic intermediate. And once you convert it to, you swap the phosphate basically from the 6th position from the 1 position to the 6th position. Then now you got the glycolytic intermediate and you're ready to go. So it's quite a few steps, actually, more than you see here. And let's look at them in detail. All right, this is what we get. We get, you drink your milk. Hopefully you have lactase. If you don't, you take it exogenously, and you break it down and you get galactose and some glucose, which goes on in its usual fate. The galactose that we get is in the beta anomer. So this is the carbon 1 right there. Okay, and there is a specific enzyme that will flip it from the beta to the alpha. Okay, so this enzyme is called galactose mutaroatase. So what is it? Obviously, it fits into this general category. We have an isomerization there. And it's an epimerase, and we've seen them before. Okay, it changes the configuration of any one carbon without affecting any other carbon. Okay, so it fits. You know, why do we call it a mutaroatase and not an epimerase, for example? Well, it's just a subcategory of the bigger group. So when we're dealing with anomers, with the anomeric carbon in the closed conformation of the sugars there, if the carbon 1 and carbon 5 have the same, R or S configuration, then we're dealing with a beta anomer. So obviously this is the case with galactose. C1 and C5 have the same R or S configuration, and now we're in the beta anomer. But what we want in this case is for the two carbons to have different, and in that case, that would become an alpha anomer. So just so we get everything straight, all these things you see here, they're all epimerases. Okay, epimerases work, however, when you have more than one center of asymmetry, and special so they change the configuration of one carbon. Racemases do the same thing. They change the configuration of one carbon, but the substrate has only one center of asymmetry, right? So all these enzymes change the R or S configuration of one carbon. That's their common thing, but then we fit them into different categories depending on other specifics. For example, mutaroatase, which is what we're dealing with, it's a specialized, it's an extra categorization of epimerases and specific for alpha and beta aldoses, which is what galactose is. How does it work? Well, the way it works is to first open it up. You open up the ring and then you close it again. And when you attack the carbonyl, after it opens, it depends on how you attack. You do the first attack, okay? So then the carbonyl will be like this, and when you attack it again, when you go to close it, okay, then you have flipped, the configuration of the carbon from the beta to the alpha here. So that's all the enzyme has to do, to facilitate the opening of the closed form of the sugar, and then attack from a particular place, from this hydroxyl group. Now the carbonyl, when it opens up to become hydroxycarbon, will be found in the alpha configuration. So that's the scheme that has been proven to be correct, and I highlight here our very own Frank Roussel in the Department of Chemistry, who was part of this work. So why bother with this? I mean, why change from beta to alpha? Okay, because the next reactions have a requirement for alpha galactose. So that's a must do before you can do anything else. All right, now what we're dealing with is fairly inert molecules. We're dealing with galactose, glucose, so they're really inert. They're not highly reactive. They're pretty stable. And how do you activate them? Well, you phosphorylate them first at the carbon 1. So we've seen fructose getting phosphorylated on carbon 1, but glucose, when it comes in, it's phosphorylated by hexokinase at carbon 6. So this is carbon 1 that gets to be phosphorylated. And then the alpha isomers, and only the alpha, undergo this nucleotidyl transfer, which we're going to spend a little more time on. Now, why activate them? Again, they're fairly inert, and the anomeric hydroxyl group is not easy to eliminate. So you have to derivatize it and make it a good living group so it can go. So just to, so that we don't get confused with the names, the enzyme that will put the phosphate on the 1 position is called galactokinase. It is not a hexokinase. Hexokinase, as we said, it phosphorylates all kinds of sugars at the 6th position, not the 1. Okay, so this is a different enzyme than hexokinases. So galactokinase will phosphorylate galactose on carbon 1. And now we have to this weird thing that we have not seen before, although we have mentioned it at the very beginning in the course. So here we're going to activate them. And how do they get activated? So remember, this is ATP in this case, adenylylation. And any one of these phosphates can be attacked. In usual phosphorylations, we're dealing with, let's say, a gamma phosphate. Here it's the alpha. That's the key one. And something, usually oxygen or nitrogen, can attack this phosphate, and the whole pyrophosphate, the beta and gamma, will leave. That's the pyrophosphate here. And what's left is the molecule that has this hydroxy, or in some cases, nitrogen group, will be attached to AMP essentially. Okay, so this is the A part, and this is adenine, the sugar, and the 1-phosphate. So that would be AMP. So the whole thing now is found on to X. So X is decorated with AMP. So in other words, X is adenylated. Right? Now the nucleotide could be anything. In fact, we will see all of them. In the next slide, we're going to see UTP being used. In other cases, we have CTP used when we activate lipids, for example. Right? So why do we do this nucleodidylations? In this case, adenylation, let's say. It is used to drive very unfavorable metabolic reactions because there is a lot of delta G here that's released. It's negative. Of course, the pyrophosphate itself, it's going to be hydrolyzed almost immediately. You get 2 phosphates, and that also adds an extra boost, let's say. So this is a way to activate things that are not really very reactive, okay? And like sugars, for example. So you spend a lot of energy currency here in the form of ATP and high energy bonds, but that's what you've got to do to use them. So if you do it on glucose, okay, glucose can be adenylated. If X now here is glucose, okay, glucose gets adenylated, and this is called glucose, and this is a form of glucose that's actually used to put into starch by plants. So that's how plants activate their glucose, and that becomes starch eventually. We do something similar with UTP when to put it into glycogen, but that we'll see in the next few videos. In this case, however, we activate glucose again with UTP. And what we end up with, okay, this is glucose 1-phosphate right there. And why do you get UDP glucose? Okay, because the 2 phosphates, okay, so this, if you imagine the U, the sugar, 1-phosphate plus 2 more, that used to be on UTP, okay? So, but when they left, because this oxygen from the phosphorylated glucose at 1 position attacked the UTP, let's go back. Imagine that this was a U here, and what you have is glucose 1-phosphate attacking this phosphate. Okay? So this oxygen on this phosphate on glucose attacks the alpha phosphate of UTP, okay, this PPI goes away, and then you have now glucose UDP, so 2-phosphates, right? So that's what you see here. Right? So this oxygen attacked this phosphate on UTP and the pyrophosphate left, and what you end up with is UDP glucose. Okay, so it's activated now. So glucose got uridylated. And what do we do with this? Okay, so that's this reaction here, the side reaction. We have our galactose 1-phosphate waiting. Okay, so we put in the wait room here. In the meantime, we activate glucose, and now the two can react. Okay, and what the enzyme is going to do, GALT, is to simply swap the two. So glucose will become glucose 1-phosphate, and the UDP now will be passed on to galactose, and we're going to get UDP galactose. So that's the reaction catalyzed by GALT, and there's a swap, as I said. And when what was uridylated, which was glucose, now becomes UDP galactose, and glucose and glucose is now left with as glucose 1-phosphate. So that's fine. How do we close the loop here? How do we go from UDP glucose to UDP galactose so that this cycle can keep going? That's a simple epimerase. It just changes the 1 carbon, carbon 4 that distinguishes galactose and glucose, and now we got UDP glucose, and the whole thing can keep going again and again when the next galactose molecule comes in, right? But from the first galactose, so from the previous galactose molecule, so now we end up with glucose 1-phosphate, which still we're not there yet. Yes, we are. It looks like glucose. It is glucose, but it's phosphorylated at the wrong position, so it's 1-phosphate. And this is what we need to deal with, and we have to convert it to glucose 6-phosphate. And these enzymes, we have seen them before. Well, it's going to be a phosphoglucomutase that's going to do it. And where have we seen them? So it's another isomerization. So it shifts the functional group from one position to another. This is the definition of mutases. And we have seen them. We have seen them at the same enzyme, similar, not the same enzyme, similar kind of reaction mechanism for a smaller substrate in the lower part of glycolysis when we change glycerol 3-phosphate to glycerol 2-phosphate. And how was it done? Well, actually, the substrate gets phosphorylated again. Okay, at the 6th position. So we have an intermediate that's phosphorylated at both the 1 and the 6th position. And then the enzyme picks up the one that was at the 1 position. So what you're left with is the one at the 6th position. That's showing here from going from right to left. Okay, this is glucose 1-phosphate, and we want to convert to glucose 6-phosphate. The enzyme on a serine is phosphorylated itself, so it passes this blue phosphate on the 6th position. So you have this intermediate now that's phosphorylated at 2 positions, both carbon 1 and carbon 6. And then the enzyme picks up the red phosphate from position 1, and the substrate is left with the one at position 6. So this is fully reversible, of course. You can go back and forth. So whenever you want to make glucose 1-phosphate, that's one easy way to do it from glucose 6-phosphate that you have a load from glycolysis. Again, the color coding helps to illustrate that it is not the same phosphate that gets moved, okay, from one position to another. There's just a swap of phosphates with an intermediate that's phosphorylated at both positions. So again, this enzyme recognizes only the alpha anomers, right? Which explains why at the very beginning, we had to convert galactose from the beta anomer to the alpha anomer. So why do we do this? Obviously, because it's a glycolytic intermediate. So that completes now the whole galactose utilization pathway, and we see how we get to utilize galactose. Why so complex, right? And this is just, I feel your pain, okay? When you go through this and you say, well, why do we need to do all these reactions? Yes, Leloir got his Nobel Prize, but why? There is actually no real answer, and people have thought about this. This is I thought I will make you feel better if you see this from an actual paper, and I highlight a few things here. Why do you have 3 separate enzymes? These are specific enzymes used for galactose utilization to change just the configuration of a single carbon. So this is a unique example. It doesn't happen in any other case we have seen in biochemistry, or we will see. Okay, the case we saw is, oh, here's an epimerase and change the carbon and we're done. Well, this is complex. Okay, so it is what it is. I guess that's very fitting for this. And if you feel confused, you're not the only one. There is no answer why this way the way it is. All right. It turns out that the GALT enzyme, which is the one that that takes the UDP from glucose and puts it on galactose, so we release glucose 1-phosphate. That's the enzyme here. This is a human disease gene. If it is mutated in people and they eat dairy products, then you have buildup of galactose in the blood, which is toxic, and that causes all kinds of problems. That's a typo here. It's galactose. An A is missing. Which, by the way, I took from the Minnesota Department of Health. They're dairy producing states. So GALT is a deficiency is a health issue. Before so this is not really sugar utilization, but it's how do mammals make lactose? Right? We need to discuss this. How do you make milk sugar at the first place? So it turns out that there is a very simple enzyme, lactose synthase. Okay, so it takes activated galactose, which again could be UDP galactose, right? Which we discussed what that is, and sticks it to glucose, and you get lactose. So it's a very simple, straightforward reaction, and we all have it. I mean, males and females and non-lactating females. Every mammal has it. Actually, many animals have it. You don't even have to be a mammal for that. The reason is that galactose modifications are common. I mean, some proteins and other molecules get galactosylated. So a galactose moiety, will be added to, let's say, to a protein, and it obviously doesn't serve any dietary purposes there, but it decorates the protein, and that's important for some extracellular proteins. For example, there is lots of or proteins on the surface of the cell that they see the outside. They're often decorated with sugars. They are glycosylated in some ways, in many ways, and galactosylation is one of them. Right. So the enzyme exists everywhere. Everywhere, meaning in males and females. But its activities is at a basal low level in most of us, right? But when a female is lactating, what happens, okay, in addition to galactosyltransferase, which can work fine by itself and modifies proteins and things like this, as I told you with galactose, now galactosyltransferase, so this enzyme here, which normally works by itself, a version of lactose synthase called galactosynthase, galactosyltransferase. Now, it is found in a complex with another protein. So this protein, lactosynthase, actually is galactosyltransferase plus another accessory protein. And that protein is called alpha-lactalbumin, and by itself has no catalytic activity, but what it does, it lowers the Km for glucose, okay? So the galactosyltransferase activity uses glucose as a substrate, extremely easy. And it is the synthesis of lactalbumin, the accessory factor, okay, that has no catalytic activity of its own, that is made in huge amount hormonally, in the mammary glands when a female is lactating, a female mammal is lactating, and that enables galactosyltransferase to become lactosynthase and use glucose as a substrate as opposed to some other protein that needs to be modified, and then you make lactose. Right? It's actually pretty cool. It's an interesting example of how an existing enzymatic activity that's it exists at the basal level everywhere. It's sort of has been co-opted, in the mammalian lineage to make this beautiful disaccharide, lactose, and pass it on to milk. All right, so we're going to stop at this point, and then the next video we're going to see how we get to utilize all these other sugars.